Hello and welcome. Have you ever wondered how scientists can teach a plant to fight off pests all on its own? Or how they can make crops survive a brutal drought? Well, it all boils down to solving one massive challenge. How do you get a brand new, custom piece of DNA inside a plant cell? Today, we're going to dive into the super clever ways scientists are hacking the code of life itself. I mean, let's just frame it this way. How do you give a plant a superpower? Because that's really what we're talking about, right? We're inserting a whole new set of instructions into its genetic code. But, unlike a computer, you can't just plug in a flash drive. You have got to find a way to get that new code past the plant's own natural defenses. And that, right there, brings us to the single biggest hurdle. You see, every single plant cell is wrapped in this tough, rigid cell wall. It's basically a microscopic fortress. So if you want to rewrite the plant's genetic software, you first have to figure out how to breach that wall without, destroying the cell in the process. So the whole collection of tricks scientists use to do this is called direct gene transfer. The name kind of says it all. These are all methods designed to sneak naked DNA, and that just means DNA all by itself, not being carried by a virus or anything, directly into a plant cell. Let's check out 3 of the most ingenious strategies they've come up with. Okay, first up, we've got the brute force method. The thinking here is pretty simple. If you can't sneak past the fortress wall, why not just blast your way through? And believe it or not, that is the core idea behind a technique that uses a microscopic cannonball. So say hello to the gene gun. It's an awesome name, right? And its scientific name is just as cool, Biolistics. It's basically a mashup of the words biological and ballistics. And yeah, it does exactly what it sounds like. It literally fires genetic material straight into plant cells. And here it is, the whole setup. Now, this is not some tiny little handheld gadget. It's a seriously sophisticated piece of lab equipment called the Biolistic PDS-1000/He system. That He at the end, that stands for helium, and that's the secret sauce. It uses a powerful, perfectly controlled burst of pressurized gas to do the firing. So check out how this works. A sudden blast of helium bursts this tiny little part called a rupture disk. That blast launches a sheet, the macrocarrier, which is holding millions of microscopic particles all coated in DNA. Now that sheet is immediately stopped by a screen, but the tiny DNA-coated particles, they keep going, flying onward at super high velocity and bam, right into the target cells. It's an incredibly precise, high-speed delivery system. So to really get it, you have to look at the ammunition. It all starts with these microscopic bullets, usually made of gold or tungsten so they don't react with anything. Then, scientists coat these tiny particles with the DNA they want to deliver. And finally, you've got the powerful blast of helium gas that provides all the force. Okay, but what if blasting your way in is just a little too messy? Well, a totally different approach is to try and create temporary doors in the wall. This next technique does just that by literally shocking the cells open. The science behind this one is called electroporation. What scientists do is apply a very brief but powerful electric pulse to the cells. Now, this shock doesn't kill them, it just creates these tiny temporary pores or holes in the cell membrane. And for just a split second, the fortress gates are wide open, and the new DNA can slip right inside. And what's really, really clever about this is that scientists can actually fine-tune the electrical shock to get different results. So a lower voltage for a longer pulse is perfect for what's called transient expression. You can think of that like running a temporary program on a computer. But a super high voltage, incredibly short pulse, that's better for stable integration, where the new DNA actually becomes a permanent part of the plant's main operating system, something that gets passed down to all its future cells. All right, so we've gone from brute force to electrical gateways. Now we come to our third and maybe the most cunning method of all, the DNA Trojan horse. This strategy is all about disguising the DNA to trick the cell into welcoming it right inside. This technique is called Lipofection, and here's how it works. You take the new DNA and you enclose it inside a tiny man-made bubble called a liposome. Now, this bubble is made of lipids, which are the exact same fatty molecules that make up the plant's own cell membrane. So because the disguise is literally perfect, the liposome can just float up, fuse directly with the cell, and deliver its genetic cargo right inside. The strategy is just beautiful in its simplicity. First, you encapsulate the DNA. Often it's a little circular piece called a plasmid inside that lipid bubble. Second, that bubble adheres to the cell and just fuses with its membrane. And finally, the DNA is delivered safely inside, having completely bypassed the cell's defenses. No force, no shock, just a perfect disguise. And this Trojan horse method has some really fantastic advantages. For one, the liposome acts like a suit of armor, protecting the precious DNA from getting broken down by the cell. It's also really gentle, with very low toxicity to the cell itself. For the scientists, it's a pretty easy procedure to perform, and it often results in great long-term stability for the new gene. So, we've just looked at 3 completely different approaches. We have a gene gun, an electric shock, and a Trojan horse, which of course brings us to a key question. Which one is the best? Well, the answer is, it depends. It's all about choosing the right tool for the job. And this comparison here shows the trade-offs perfectly. The gene gun, its huge advantage is that it works on intact tissues. You don't have to remove that tough cell wall first. The downside, while the equipment is really expensive and the process can actually be a bit harsh and damage the DNA. Electroporation, on the other hand, is simple, it's fast, and it's cheap. But its big drawback is that the prep work can be really laborious, often requiring you to get rid of that cell wall first with enzymes, which is a super delicate and time-consuming process. So, at the end of the day, there is no single best method. There's no one-size-fits-all solution here. A scientist's choice is going to depend entirely on the specific plant they're working with, the lab's budget, and of course, what the ultimate goal is. Are they looking for a temporary change or a permanent one? These tools, this whole toolkit, gives us an absolutely incredible power to rewrite the code of nature. We can design plants that grow in salty soil, or plants that produce more vitamins, or even plants that can create vaccines. And that leads me to a final question for you to think about. Now that we can write new code for plants, what should we ask them to do next?